Hi everyone, hope you're ready for yet another fruitful lesson. I am here to take you through using an eyepiece graticule and stage micrometer to make measurements. So this is a concept that is going to help you in your practicals and generally in your papers. You'll be expected to know how to use an eyepiece graticule to make measurements. So how is an eyepiece graticule used? When you look at cells, as you study them using a microscope, remember that a microscope has an eyepiece. An eyepiece is where you place your eye so that you view what is inside the microscope, so that you view the specimen. So someone could be interested in measuring cells at the same time when they are looking at them in a microscope. Remember that you cannot get a ruler and you place it in the microscope so that you're able to measure the size of the cell. That will not be possible. So there is a small instrument known as an eyepiece graticule. So this eyepiece graticule is a transparent scale. Assuming this is your eyepiece and this is your field of view. So usually that transparent scale is going to be, most of the time it's inbuilt within an eyepiece. So some eyepieces have already inbuilt eyepiece graticules. That is why it is called an eyepiece graticule, because it's formed within an eyepiece. And this is how it looks like. It has small divisions. You can see these divisions. Those divisions are called eyepiece graticule units. Most of the time, the divisions run from 0 to 100. 0 to 100. Now, from 0, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Those are 10 divisions. And it goes on up to 100. So assuming you are using an eyepiece that has a graticule and you're observing cells. An example, let's look at this diagram. It's going to bring it out. Assuming this is what you're seeing through the microscope, you're observing cheek cells, cheek epithelial cells, and these are the cells you're seeing through a microscope. So you would be interested in finding out the size of maybe one of the cells. And the eyepiece graticule is going to be used to help you determine the size of any of the cells. So when you look at all these cells, it is always better that you choose a cell that is well aligned with the eyepiece graticule. And if you look at all these cells, this is the one that is most well aligned. Yes, you can consider this one, but you can see this is well aligned with the eyepiece graticule. So it's the one that you should go with. Yes. And after you have chosen the cell to measure, what is the next step? The next step is to identify, when it comes to this cell, how many eyepiece graticule units? These eyepiece graticule divisions can also be called eyepiece graticule units. So how many eyepiece graticule units is this cell taking up? You can clearly see that the cell, when you look at the position in which it starts at the 40th division and ends at the 60th division. So obviously, for me to know the size of this cell, I'm going to consider the divisions the cell is taking up. That is always the first step. How many divisions or how many eyepiece graticule units is the cell taking up? So here we are just going to get the difference between 40 and 60. And when you look at the difference, we can clearly see that those are 20 eyepiece graticule units. I'm just writing this as an abbreviation. 20 eyepiece graticule units. So this cell is equivalent to 20 eyepiece graticule units. Sometimes you might need to count the divisions individually. So this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. But the easiest way is just look at where is it starting from or is it ending, get the difference. Those will be the number of divisions that the cell is taking up. So that is the first step. So how do we get to know the size of the cell? Now, the measurement we have made, we have just counted units. They are just units. But remember, cells are measured in either millimeters or micrometers. But here I'm just having eyepiece graticule units. So our next step is to calibrate the eyepiece graticule. What does calibration mean? We are going to get these divisions and turn them into known units, maybe millimeters, maybe micrometers, maybe nanometers. So let's go to the next step and see how we are going to be able to calibrate the eyepiece graticule. Okay, just a recap, the cell lies between the 40 and 60 division on the 60th division on the scale. So that means that our cell measures 20 eyepiece units in diameter. 20 eyepiece units in the diameter. So the next step is to find out what are these 20 eyepiece units equivalent to in terms of millimeters or in terms of micrometers. So that takes us to calibrating this eyepiece graticule. So for us to calculate the eyepiece graticule, we are going to use a very tiny miniature transparent roller called a stage micrometer. So that stage micrometer has divisions, but the divisions are of known units. That is the difference. Remember, the eyepiece graticule was just divisions, We don't know whether they are in millimeters, they are not in micrometers, they are not in nanometers. But when it comes to the stage micrometer, for its divisions of particular units that are known. So you find that maybe the divisions are in terms of millimeters. Like here they have labeled this stage micrometer. And they are showing that from this point to this point, that is equivalent to 0.1 millimeters. It can vary. It could be 0.01 millimeters or 0.1 millimeters. But according to this, the stage micrometer is equivalent to 0.1 millimeters. Then from this section to this section, still is 0.1 millimeters. And then you go on. So what you're going to do, you're going to get the eyepiece graticule and align it, superimpose it very well with the stage micrometer. So we are going to use the stage micrometer to calibrate the eyepiece graticule so that we know what is one eyepiece graticule equivalent to in terms of millimeters, because our stage micrometer is in terms of millimeters. So in this case, you can choose any section to work with from this diagram. Let me give you an example. An example someone could consider using this section from here to this section. So in that case, we can see that 0.1 millimeters are equivalent to how many eyepiece graticule units? You can see that. We can see that 0.1 millimeters are equivalent to 40 eyepiece graticule units. Oh, that word, eyepiece graticule units. Or maybe let me just write units. Sometimes the words are too long. 0.1 millimeters are equivalent to 40 units of the eyepiece graticule. Now, remember the cell we measured the other side was equivalent to 20 units. So I can do the math and be able to find out what these units are, and I will be able to turn them into millimeters. So that is going to be simple. You do the math, 40 units are equivalent to 0.1 millimeters. How about the 20 units that I measured? How about the 20 units? So here I'm trying to turn units into millimeters. So the 20 units are going to be equivalent to 20 times 0.1 divided by 40. But remember, your answer is going to be in millimeters. Your answer is going to be in millimeters. If you do the math, 20 times 0.1 divided by 40, you're going to get 0.05 millimeters. That means that the size of the cell is 0.05 millimeters. But remember, the base unit of measurement, that is most of the time a device to be used, is micrometers. So turn these millimeters into micrometers. Remember, we said that 1 millimeter contains 1,000 micrometers. So how about the 0.05? Whenever you are writing your answers, please show the working. It's going to be 0.05 times 1,000. And if you place that into a calculator, it's going to be 50. It's going to be 50 micrometers. So that is for someone who considered this section from here to here. Another person could decide to consider another section. An example, if you look at this from here to here, we have 0.1 millimeters. From here to here, we have 0.2. So someone could be like, okay, 0.2 millimeters are equivalent to 80 units. If you do the math, you still arrive at the same answer. 0.2. Okay, let me start with the units since we are changing from units to millimeters. So you could write that 80 units are equivalent to 0.2 millimeters. How about the 20? How about 20 units? If you do the math, you're going to find that the answer is going to be the same. 20 times 0.2 divided by 80. The answer will still be 50 micrometers. Because the answer will be 0.05. And then you turn it to micrometers. It will still be 50 micrometers. Another person could decide that, okay, when I look at the whole length of this stage micrometer, from this side to this side, that is 0.25. So you do the same math. 0.25 millimeters are equivalent to 100 units. So how about that? You still arrive at the same answer. So whenever you are using an eyepiece graticule to measure an organelle or a cell, always, first things first, know how many divisions of the eyepiece graticule is that cell or organelle that you're measuring taking up. Then after that, calibrate the eyepiece graticule using a stage micrometer. Let's look at examples. We are going to look at several examples of where this is applied. Okay, we have already done this. So this was our answer, 50 micrometers. We have a question here. Figure 2.2 is a photomicrograph of a stained transverse section through a root of a different type of plant. So we are having a root of a plant And there is a section in the middle of the root. That section, okay, that is the vascular tissue. It's running from line X to line Y. And they are telling us that one eyepiece graticule division is equivalent to 24.6 micrometers. What is the size of the line XY? You can see that this line XY is well superimposed or is well aligned with the eyepiece graticule. So this is the eyepiece graticule. Remember, the eyepiece graticule has those small divisions. So what I'm going to do, I'll get a ruler and align this point X to see what it is matching with this side, and then even this point to see what it is aligning with this other side, and then count how many divisions are there in between. If you look critically, you'll see that the line X is well aligned with the, because if this is 0, that means this is 1, 2, 3, 4. That is the 4th division. So the line is running from the 4th division to, then you also come this side. So this is 20, this is 25, 26, 27, I think 28. 4th to 28. So get the difference between the two. You're going to find that your answer is going to be 24 divisions. So it's taking up 24 divisions. If you want, you can count the divisions individually. You count them 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. But the easiest thing for me that I usually advise is just find what is that first division that is the line or whatever they are telling you to measure is well aligned with. Like in this case, we saw it was the 4th. And then this side, it's at position. It's at the 28th or 27th. 27th or 28th. But you just have to make sure that you are more accurate. So I have taken the 28th, and when I get the difference, I'm having 24 divisions. So that is the first thing. Measure line X, Y, get to know how many divisions it's taking up. So it's taking up 24 divisions or 24 eyepiece graticule units. Remember that divisions can also be called eyepiece graticule units. So they have already told us that one eyepiece graticule unit or division is equivalent to 24.6 micrometers. So here they had already done the calibration for us. Then how about the 24 divisions? So you just had to multiply, which means the answer was supposed to be 24.6 times 24. And then you get your answer and you write it down. 24.6 times 24. So the answer was 590.4 micrometers. That will be the answer. So that is all you have to do. Let's look at another example so that we really get this into our heads. Let's look at another example. If you look at the guide to this question, you're told that the student should be able to count and record number of eyepiece graticule units, divisions or units for line XY. Record them, count them and record. Then show the number of eyepiece graticule division units for line XY multiplied. We multiplied. And then you wrote the right answer. And then make sure that your answer has units. That is where the marks are coming from, and the question was taking 4 marks. So whenever you find questions that require the working, please make sure that you show each and every detail of the working because we don't want to lose a mark. Most of these calculation questions, I look at them as free marks, marks that you can easily earn compared to you finding a question they ask you to explain and maybe you can't even start to explain. So these calculations should always be for us to get. Let's take a look at another example. Here we are having a diagram showing a graduated slide with divisions of 0.1 millimeters. Now, when I find such a diagram, in this case, we are having two things as someone is observing through a microscope, assuming this is the aerial field of view of the eyepiece. You can see that this eyepiece has two things within it. It has this upper scale and then this lower scale. So the one that always has very tiny divisions, that is always the eyepiece graticule. So that is the eyepiece graticule. The one with the larger divisions, is always the stage micrometer. So this is eyepiece graticule. Then this is a stage micrometer. A stage micrometer. So when I look at this, actually one of the things I can easily do is first of all to calibrate the eyepiece graticule using that stage micrometer, which is well superimposed with the eyepiece graticule. And they have told us that the diagram shows a graduated slide with divisions of 0.1 millimeters. Now, whenever I see these divisions, which they are mentioning about, but they have units, I'll know these divisions are for the stage micrometer because it's the one that is usually having units all calibrated already. So I know they're telling me each division is 0.1 millimeters. That means that if I consider this division to be 0, the next will be 0.1 millimeters, and then the next will be 0.2 millimeters. That means that between each division, the size is 0.1 millimeters. So after noting that, can't I calibrate this eyepiece graticule? I'll still consider any point. In this case, I can decide to consider this section from here to here. So I can see from here to here, those are 50 divisions because it's starting from 0 up to 50. That means that 50 units or 50 eyepiece graticule units or 50 divisions of the eyepiece graticule are equivalent to, you can see that 50 is well aligned with the 0.1. So that is equivalent to 0.1 millimeters. It's equivalent to 0.1 millimeters. So this calibration is going to be very important in our next section. Or someone could consider this section from this side to here, from 0 to 100, and they're like 100 units are equivalent to 0.2 millimeters. You just choose what is easier for you, but just always ensure that whatever you're choosing, those divisions are well aligned with the stage micrometer that you are using. Okay, so next, let's look at what the question is asking for. Let's look at what the question goes ahead to ask. So they're giving us some information here that pollen grains were grown in a sugar solution and viewed using the eyepiece graticule. Remember, this is a continuation of the previous slide. Using the eyepiece graticule, Diagram 1 shows the pollen grains at the start. Diagram 2 shows the pollen grains after 4 hours. So this is diagram 1 showing you pollen grains. And still you can see these. Remember the divisions, those tiny divisions, that is an eyepiece graticule. Then here, this is still an eyepiece graticule. So we are having two situations, looking at pollen grains before growth of pollen tubes and even after pollen tubes. But growth of the pollen tubes took 4 hours. What is the question? So the question is asking, what is the length of the pollen tube in micrometers? So I'm having the pollen tube. So the question is, which one is the pollen tube? You can see the pollen tube is running from here to here, or from here to here, or from there to there. But you have 5 pollen grains. So which of the pollen grains can you see that is well aligned with your eyepiece graticule? So do not just choose any random organelle or any random structure that you're going to measure. No. Always look for the one that is well aligned with the eyepiece graticule. And in this case, this is the most appropriate candidate. So all you have to do is get a ruler. Please never go into a paper without a ruler, not even a practical, not a paper 1, multiple choice, or paper 2 structured. Always have your set with you, a ruler and your calculator. Always be ready. Okay, so you're going to get your ruler and you draw a line that is going to be well aligned with a point here on the eyepiece graticule. And we are measuring the pollen tube. The pollen tube is from this region to this region. Do not include this. This is just the pollen grain, but we are considering the pollen tube. So I also have this region from here down. So I can count how many divisions are in between. When I look at this, this was at the 24th position. So it's running from 24 to 35. To 35. So I can get the difference between 35 and 24. When you get the difference, mathematicians, what do we get? 35 minus 24. Those are 11 units. Meaning the pollen tube is equivalent to 11 units. But they're asking me, what is the length of the pollen tube? So I have to turn this into known units. And they were very specific in micrometers. But let me take you back to our previous slide. Remember, in our previous slide, we had already calibrated the eyepiece graticule. We had already calculated the eyepiece graticule. If you go back this side, you could see that the eyepiece graticule, we saw that 50 divisions or 50 units were equivalent to 0.1 millimeter. So that is what I'm going to use. I've seen that the pollen tube is equivalent to 11 units. 11 units. And from the other side, we have seen that 50 units were equivalent to 0.1 millimeters. So how about the 11 units? So you do the math. So that is how you're going to cross multiply. So you're going to have 11 times 0.1 divided by 50. But those are millimeters. So 11 times 0.1 divided by 50. What do we get? We get 0.022 millimeters. But the question was asking for the length of the pollen tube. This means this is the length of the pollen tube, but they want it in micrometers. They want it in micrometers. And we already know that one millimeter, we already know 1 millimeter is equivalent to 1,000 micrometers. So how about my 0.022? So that means you just had to cross multiply still, so that you have your 0.022 times 1,000. And that is how our answer is going to be. So multiply that by 1,000. That is going to be 22. Yeah, that is how our answer is 22 micrometers. That means that the length of this pollen tube is 22 micrometers. 22 micrometers. So that is the math you are expected to do. You can even find another question following this. Let's look at another question following. So the other question is asking, still we are considering the same pollen tube. They're asking, what is the growth rate? What is the growth rate of a pollen tube? I might even never have taught you about calculating rate, but you find it in a question because most of these questions are for application. You should be able to get knowledge from different fields and then you integrate it. Like here we have been looking at just calibrating an eyepiece graticule, but now they're asking us for the growth rates. Remember, whenever we mention about rates, time comes into mind. If you look at this pollen tube, it took 4 hours for it to grow. So that means, excuse me. So that means we are going to get the size, length of the pollen tube and divide by the time. So when we do that, it's going to be, the answer is going to be 22 divided by 4. But remember, you're dividing micrometers. You're dividing them by hours. So you can see here, when you look at these possible answers, we have millimeters per hour, meaning this won't work because our answer is in micrometers. This will also not work. So I'm left with B and A. So 22 divided by 4, what we get? 22 divided by 4. You get 5.5. 5.5 micrometers per hour. When you look at B and A is the most appropriate. That means I will take A. That means I will take A. So that is how we use an eyepiece graticule. This knowledge is very important for your practicals and your other questions that you find in either the multiple choice or the structured paper. So this is another question. They are still giving you the same information. The diagram shows a stage micrometer with divisions of 0.1 millimeters. Still, we are going to go through this so that you become very familiar with how to use an eyepiece graticule. So remember, I've told you that whenever you see a setting like this lower one is the eyepiece graticule. Why? Because of those very many divisions. The upper one is the stage micrometer. So what next? Calibrate the eyepiece graticule using this information. They are telling me the stage micrometer has divisions that are 0.1 millimeters apart. That means if I consider this to be 0, here I'll have 0.1 millimeters, and then still I'll have 0.2 millimeters. That is what I'll be having. So I can do the calibration. So in this case, I can see from the 10th division to the 50th division, the distance apart using the stage micrometer is 0.1 millimeters. So 50 minus 10, you get 40 divisions in between there. So that means that 40 units or 40 eyepiece graticule divisions or units are equivalent to 0.1 millimeters. Okay, so I'll note this because this is going to be very important as I answer this next question. Or you could be like, we have from 10. I'm counting from 10 because this region is well aligned. I might not really consider this part here. I'm going to consider this section going this side. So someone could consider from the 10th to the 90th division. The 10th to the 90th division, the difference there is 80. 90 minus 10, you get 80. Someone can still use the same, that 80 units are equivalent to, you can see 80 is well aligned with 0.2. From here to this side is equivalent to 0.2 millimeters. You just choose what to use. You use what is easier for you. Yeah, you always have that choice. So let's go to the question. The question is asking, what is the area of field of view of the microscope at this magnification? Area of field of view. So they're asking for the area. This is the field of view. So they're asking for the area. Remember now, this is a circle. I might even have never taught you about how to get the area of a circle. But you have learned from your primary, secondary. I'm sure every one of you should know how they get an area of a circle. Area of a circle is pi r squared. I have the pi given to me. The pi is 3.14, but now I need the r. So I need the radius. I need the radius. So how am I going to get the radius? You look at the middle of this circle. You can see that the middle of this circle is taking up how many eyepiece graticule units? We can see that if I'm to look at the middle of the circle, it's running from 0 to 50. That is where the middle of the circle is. So that means this is our radius. So the radius is equivalent to 50 eyepiece graticule units. You can see it very well. 50 eyepiece graticule units. I already know that 40 units are equivalent to 0.1 millimeters. So can't I use this to find out what the 50 units are equivalent to? So I'm going to use that 40 units are equivalent to 0.1 millimeters. How about the 50 units? We are trying to find out the radius. So it's going to be 50 times 0.1 divided by 40. But these are millimeters. Those are millimeters. So 50 times 0.1 divided by 40, we get 0.125 millimeters. But when I look at the possible answers, there is nothing like millimeters. That means I have to change to micrometers. Remember that 1 millimeter equivalent to 1,000 micrometers. So how about the 0.125? Do the math. We are going to cross multiply, which is going to be 0.125 times 1,000. So when you do that, our answer is going to be times 1,000. Our answer is going to be 125 micrometers. So this is our radius. That is our radius. So can't we use the formula? Our formula is area over circle is equivalent to pi r squared. Our pi is 3.14. You can see here in the answers, the pi is still there. And if I look at the answers, I might not even go even ahead to find. Anyway, let's first feed it in. So we shall have pi. Our r is 125 times 125, but all these are in micrometers. So if you look at all these, this won't be a possible answer because they're having 12.5. This won't work. This won't work. You can see now, according to this option, this is the best. According to the options given, this is the best option. Pi r squared. So our answer is C. So always look at the answers that have been given, especially for multiple choice. They give you an idea on which ones to eliminate and which ones to remain with. They give you an idea on the steps you have to take. Like you could see all this was micrometers. That means we had to change our millimeters to micrometers. And then you could see that the pi was still existing. That means you don't have to first carry out the calculation because the pi is still there. That's why I didn't insert there the 3.14. So that is our answer. As you do these questions, remember that application is very important. You'll find questions on some things that a teacher might never have mentioned about. Maybe they ask you for the area of maybe a cylinder or volume of something, but all you have to do is use the information you have gained, the knowledge you have gained over the course, and then be able to answer the questions. That means that you have to be able to think outside the box. But the beauty is that the more practice we do, the better we become. So I always encourage you to practice. If you need one-on-one lessons, I do provide one-on-one lessons at a cost. So you can get in touch with me. My Gmail is in the description box, sharukshansi@gmail.com. If you need one-on-one lessons, if you have questions that you need me to help you answer, or if you need me to do revision with you, I also provide all those services. So don't hesitate to contact me. And that is basically what an eyepiece graticule is about. This is another question. This is going to be the last question that I'm going to help you do. If you can pause and first try it out on your own so that you find out whether you can answer the question. Yes, you can pause and first try it out on your own. Okay, so the question shows us still a stage micrometer. Remember, first things first, always note. This is eyepiece graticule. This is stage micrometer. I'm just going to write stage. They are telling us the divisions of the stage micrometer are 0.1 millimeters apart. Still, I always use an example, assuming this is 0. Here we shall have 0.1 millimeters. Then here we shall have 0.2 millimeters. Calibrate the eyepiece graticule there and then consider any point. I'm going to consider the point from this section to this section, from 10 to 50, from the 10th unit to the 50th unit. The difference there is 40. That means that 40 units. Someone could be wondering, will it always be 40? No, please always first look at the diagram given to you. Don't take it like a guarantee that it will always be 40, because in the previous examples we've looked at, it was 40. 40 units were equivalent to 0.1 millimeters. So it's not always a must that that will be the case. Yeah, so 40 units are equivalent to 0.1 millimeters. Once I have this, then I can proceed with the question. So they're telling us the same eyepiece unit is now used to examine a blood smear. How many graticule divisions will cover the diameter of a cell that is 10 micrometers? So they are giving me the size of a cell. A cell is 10 micrometers. Now this means I have to change this 0.1 millimeters to micrometers first, so that I use the same units. So for me to do that, remember that 1 millimeter, we just get back to, we are changing this to millimeters, sorry, to micrometers. We know that 1 millimeter is equivalent to 1,000 micrometers. How about the 0.1? To be equivalent to, you cross multiply. Do the math, 0.1 times 1,000. And the answer is 100. So I'm having 100 micrometers. Remember, these 100 micrometers are equivalent to 40 units. So they're telling me there is a cell that is 10 micrometers. How many units is it going to contain? I'm going to use this to know how many units that cell contains. So 100 micrometers, I can write it the other way around, are equivalent to 40 units. How about the 10 micrometers? So that means I'm going to have 40 times 10 divided by 100. And the answer is 4 units. That's how you get the answer to be 4. Yes, that's how you get the answer to be 4. So please go to those past papers, try out as much questions as possible. Remember, the better you practice, the better you become at a concept. So try out as much questions as possible. Otherwise, we can call it a day for today's lesson. That was it about an eyepiece graticule. And see you on the next one. Thank you.